Welcome to the last session of the SPIM 2024. This last session is focused on aging and any kind of concept that could be relevant for aging, including cosmetics, but also dermatology. I know that there is some people coming from cosmetics, so all of us probably discussed in your own corporate management about what could be done based on longevity rules. And I know that it's really a new thing for all of us, all of you. So this afternoon we are so lucky to listen to Professor Nektarios TAVERNARAKIS. So if I can introduce you, Nektarios, thank you so much for coming in the small SPIM. I know that this year you participate to 28 Congress as invited speaker. So we appreciate that you come for us. Nektarios is probably one of the most experts all over the world on mitophagy. And I'm really curious of the global of your presentation. Just to introduce, there is too many things to exchange with people, but just to simplify, so Nektarios is chairman of the fourth director group in Greek. He's also professor in the medical school at the University of Heraklion. As I mentioned, just to understand and to know and to have an overview of the contribution of Nektarios on genetic approach in longevity, in aging, neuroaging, and all over the world. 28 contribution keynotes of Congress this year. Chairman of the European Institute of Innovation and Technology, elected member of the European Academy of Science, elected fellow of the Royal Society in London, elected fellow of the American Association for Advancement of Science, elected member of the German Academy of Science, and also the European Academy of Physiology and Molecular Gallium Prize Award. Thank you so much for coming.
Thank you, Professor Breton. Thank you for inviting me. It's an honor for me to be here this afternoon. I would like to thank the organizers as well, especially Elizabeth, because she's the reason I made it today. I had an adventure with my flight coming from Athens. It was delayed because of the snow, and then the flight to Vichy was canceled. But she managed to find a way to bring me here, and made it everything was so smooth, and I didn't really have to do anything. So thank you for all the help. I appreciate it. And this gives me the opportunity to be here and present some of the work that we are doing related to mitochondria, mitochondrial homeostasis, and more specifically the mechanisms that are involved in regulating the number of mitochondria in cells. Before I begin, let me just give a brief introduction and also make a connection with skin physiology. So of course we know that mitochondria are generators of ATP. They generate the energy that cells need to sustain themselves and perform their functions. They are also, in addition to ATP, generators of toxic byproducts of metabolism called reactive oxygen species, the famous ROS. Now, of course, this is a process that cannot be avoided. It's an intrinsic source of damage for mitochondria, but also for cells. If we were to eliminate ROS, we would essentially need to shut down mitochondria. So this is something that we cannot afford. Actually, it has been calculated that about 1%, up to 2% of oxygen that we consume is converted to ROS in mitochondria. And this, of course, results in damage, and the most proximal targets for this damage are mitochondria themselves. So they sustain damage through their functional life. And this can be summarized as a phenomenon of aging for mitochondria. So they start their lives as healthy, functional mitochondria. They produce a lot of ATP and very little ROS. But during the course of their lifespan, they deteriorate, and eventually we end up with mitochondria that are almost non-functional. They produce mostly ROS and not ATP. And of course, because of this, mitochondria dysfunction has been implicated in many, many pathologies associated with skin physiology and pathophysiology. So, for example, exposure of the skin to pollution, UV radiation, and other environmental factors will result in mitochondrial dysfunction, among other things, of course. And here, of course, cells have a way of replenishing their mitochondria if mitochondria become damaged, either because of intrinsic reasons, like ROS, produced by mitochondria themselves, or extrinsic reasons, toxins, exposure to UV, radiation, etc. Cells have a way of replenishing their mitochondrial pool. Now, a very important sign of pathology, and actually a common denominator of all these diseases, is accumulation of damaged mitochondria. Now, this accumulation of damaged mitochondria is seen in the context of disease, but is also seen in the context of physiological aging. So not in the context of any pathology, but during physiological aging, what has been observed, and actually in very different organisms, ranging from mammals to C. elegans, to the nematode C. elegans that is used in the lab as a model for studying aging. We see this accumulation of mitochondria. So this is the rat ventricular tissue here. We can even quantify this accumulation of mitochondria during aging. If we compare rats of 6 months of age to rats that are 24 months of age, there is a clear accumulation of mitochondria. And then in C. elegans, we can even follow this phenomenon of mitochondrial accumulation during aging in vivo, in live animals. We don't need to sacrifice them and perform electron microscopy. Here we can just label mitochondria using GFP and we use that as a proxy to evaluate, to assess mitochondrial content. We can even do this in a cell-specific manner if we use specific promoters to drive these mitochondrial GFP reporters in specific tissues. In this case, this is the intestinal tissue of the animal. And if you compare day 1 to day 10, there's a very clear accumulation of mitochondria, so the mitochondrial number is increasing. And this is already quite old for these animals that live for about 15 days. So comparing day 1 of adulthood to day 10, there is an accumulation of mitochondria. So this was intriguing because, of course, there has to be a molecular mechanism that either fails or brings this about. And we wanted to study this phenomenon of mitochondrial accumulation that is a common denominator both in disease and in physiological aging. And what actually made it even more interesting for us is the following observation that was made by a very talented postdoc in the lab some years ago. So what she noticed is that actually in long-lived animals, and perhaps you know that in certain model systems, such as C. elegans, we can even manipulate aging. We can extend the lifespan of these animals by introducing specific mutations in genes, for example, that control insulin IGF-1 signaling or TGF-beta signaling. Now, these mutants are long-lived. They live much longer than wild-type animals. And what Marta observed is that although wild-type animals accumulate mitochondria during aging, these long-lived mutants don't accumulate mitochondria. So this suggested, it's a piece of evidence that there is a connection between the number of mitochondria in cells and longevity. And perhaps having too many mitochondria or allowing a mitochondria to accumulate is detrimental, is shortening lifespan. Now, of course, to really address this question, one would need to be able to manipulate mitochondrial content at will and then examine what the consequences are on aging. And this is something that we can do in C. elegans, and that's why this is a very advantageous model to study aging, because it's genetically malleable, and you can introduce mutations to specific genes that will actually alter mitochondrial content. In this case, we introduced mutations in 2 mitochondrial genes called prohibitins. Now, these proteins, if mutated, result in the accumulation, in the further accumulation of mitochondria in wild-type animals. So they make things worse. in wild-type animals. However, if we do the exact same manipulation in long-lived mutants, we further suppress this accumulation of mitochondria during aging. And now we can ask, what is the consequence in lifespan? And indeed, what we see is that in the case where we made things worse, we accelerated this accumulation of mitochondria during aging. These animals lived shorter periods, so their lifespan was shortened. However, whenever we even further lower this accumulation, we extend lifespan of these animals. So this suggests that actually there is a causative relationship between the number of mitochondria, this phenomenon of mitochondrial accumulation, and longevity. If there is mitochondrial accumulation, then we have shorter lifespan. If we block this accumulation, then we extend lifespan. Now, this is an observation, of course, but it doesn't tell us anything about the mechanism that is underlying this phenomenon. So what are the mechanisms that regulate mitochondrial content in aging? If we think about it, we have a phenomenon of mitochondrial accumulation. Two things might be happening. There are two processes involved: mitochondrial biogenesis, that generates mitochondria, and mitochondrial degradation, that recycles mitochondria, degrades mitochondria. So these two processes, normally under physiological conditions in young cells, are balanced. So we have mitochondrial biogenesis on one hand, we also have turnover of mitochondria on the other, and there is a steady number of mitochondria in cells. Of course, cells can adjust these two processes. Depending on energy needs, they might increase their mitochondrial number or decrease it according to physiological conditions, nutrient availability, etc. But balance will always be restored. Here, what we are talking about is something different. What we see during aging, and also in age-associated pathologies, many of them affecting skin, we see a collapse of this delicate balance, and instead we see runaway accumulation of mitochondria. So what is failing? What is happening in cells that allows this runaway accumulation of mitochondria? So that is the question. That's what we wanted to address. And to do this in a genetic model, we can perform genetic screens for genes that affect this particular process. So we devised the strategy to isolate mutants that affect the balance of mitochondrial content in cells. I'm not going to go into the technical details of how the screen was done in the interest of time. I'm going to just introduce 3 genes that were recovered out of this screen, among others, of course. DCT1, NIX, PINK1, and parking. All these 3 genes are involved in a process called mitophagy. Now, what is mitophagy? Mitophagy is an autophagic process, as the name suggests, but it's a cargo-selective autophagy. It targets mitochondria specifically. So autophagosomes are formed around mitochondria, and these mitochondria are delivered to the lysosome for degradation. This process of mitophagy was first described in yeast as a physiological response in the absence of food. So when yeast cells don't have nutrients, they activate mitophagy to eliminate most of their mitochondria because they don't need them. There's no food and there's nothing to burn. And instead, recycling mitochondria provides nutrients for these cells to survive under conditions of starvation. Now, there are, of course, other physiological functions of mitophagy. You might be aware of mitophagy in erythrocytes. Here, erythrocytes, in addition to not having a nucleus, also don't have mitochondria. Red blood cells don't have mitochondria. They are eliminated through a specific mitophagy process involving NIX, which is the homologue of DCT1 that we identified in C. elegans. This protein NIX is a so-called mitophagy receptor. It is actually a protein localized on the outer mitochondrial membrane and interacts with LC3, an autophagosomal protein, and this directs the formation of the autophagosome around the mitochondrion. And that's how mitochondria are recognized and eliminated. And this, of course, has been evolved in this way because we don't want mitochondria in red blood cells. Red blood cells carry oxygen, they transport oxygen, and we don't want the organelles that consume this oxygen in those cells. So that is why this is a physiological process of mitophagy, eliminating red blood cell mitochondria. Now, there is also a pathological paradigm, and this involves the other two players I talked to you about earlier, pink and parking, which are involved in the recognition of damaged mitochondria in cells. So whenever a mitochondrial becomes damaged beyond a certain threshold, it is recognized by this pathway because PINK1 is activated locally in the vicinity of the damaged mitochondrion by signals emanating from the damaged mitochondrion, for example, ROS or calcium leaking out of mitochondria. This locally activates PINK, which then locally phosphorylates and activates parking. Parking is an E3 ubiquitin ligase that ubiquitinates specific proteins on the outer mitochondrial membrane. These proteins are mitophagy receptors. And by analogy to this paradigm, that's how autophagosomes are formed around mitochondria and eliminated. But in this case, we have selective elimination only of damaged mitochondria. So cells indeed have a way of recognizing damaged mitochondria and selectively removing them without actually damaging or degrading healthy functional mitochondria. This is very important, to have a mechanism of selecting which mitochondrion to eliminate. It is really very important for health survival and physiological functions. Now, this also is a conserved pathway in C. elegans. As I said, we identified the nematode homologs of these three players. We further characterized the ubiquitination events on DCT1, which is the homolog of NIX. This protein is ubiquitinated in a specific lysine residue, and this is a trigger for mitophagy. That is how mitophagy is upregulated in C. elegans. And this, because we now have a bona fide specific mitophagy receptor, this allows us to monitor this process in vivo, something that cannot be done, for example, in the mouse or in other models. Here we can use fluorescent reporters to actually monitor mitophagy in vivo. And we developed two systems. One is based on the co-expression of mitochondrial reporter. So this labels mitochondria, and another labels autophagosomes. So whenever an autophagosome engulfs a mitochondrion, then we have colocalization of these two dyes, and we can see this very easily. And actually, this is what we see under conditions of stress. So under physiological conditions in wild-type animals, we have very little colocalization between these two reporters. But under conditions of low insulin signaling, as you can see, heat stress or CCCP treatment, which is an uncoupler of mitochondrial respiration, we see extensive colocalization of mitochondria and autophagosomes, signifying the induction of mitophagy. So this is one reporter. There's also another more convenient and versatile reporter that is based on a bipartite fluorescent reporter construct. This is based on rachiometric imaging. So here we fused GFP and DS-RED and direct the expression of this reporter to mitochondria. Now, GFP, as you know, is quenched, the fluorescence is quenched by acidic pH. So whenever mitochondria find themselves in lysosomes, which is the final step of mitophagy, that's where mitochondria are degraded in lysosomes, then GFP fluorescence drops. DS-RED, on the other hand, remains steady. So that is how we can quantify. In this case, we can even quantify. This is not just qualitative. We can even quantify the levels of mitophagy induction. And whenever mitophagy is induced, then we have a drop in the ratio of GFP versus DS-RED. For example, if you compare wild type to DAF2, insulin signaling deficient mutants, there is this drop of fluorescence, signifying induction of mitophagy. Same thing with CCCP treatment, heat stress, or paraquat, another inducer of heat stress. So now we have a tool that allows us to monitor in vivo mitophagy in various tissues. And this allows us, of course, now to ask what happens if we specifically downregulate mitophagy in specific cell types, away from other types of autophagy. Because if we use a general autophagy regulator, such as Becklin, for example, then we affect all types of autophagy, including mitophagy. But if we want to dissect the contribution of mitophagy specifically in cellular physiology and aging, we need a way to be able to block mitophagy specifically. And having a mitophagy receptor allows us to do exactly that. And this is what we did here. In wild-type animals, if we if we downregulate DCT1, which is a mitophagy receptor, under normal conditions, we don't see any change in the lifespan of these animals. This, on one hand, might be expected because under physiological conditions where there is no stress, there is no significant mitophagy, so not having a mitophagy receptor might actually not have an effect. But it is paradoxical to some extent, and I will explain later. Now, in conditions where mitophagy is actually induced and plays a role in the long-lived DAF2 mutant here, these animals are long-lived, if in those animals that also show activated mitophagy, this is what I showed you in the previous slide, if we now downregulate the expression of a mitophagy receptor, DCT1, we take away part of their longevity. So we make them live a shorter period of time, and this part is what is contributed by mitophagy. Of course, there are other reasons why these animals are long-lived, but one main reason is mitophagy, because if we lower mitophagy, we take away part of the longevity of these animals. And same thing, of course, if we downregulate PINK1 or parking in this particular case. Now, what are the consequences of mitophagy downregulation in cells and tissues? What we observe is that there is accumulation of mitochondria, and actually this is more prominent in hypodermal and epithelial cell types. This is what we see in C. elegans, but it has also been confirmed in other models, such as Drosophila and mice. So epithelia, the hypodermis of animals, are particularly sensitive to mitophagy downregulation. In addition, we don't just have accumulation, we don't just have more mitochondria in these cells, but the whole mitochondrial network becomes misshapen and disorganized. So for example, in this case, we observe muscle cells. Instead of having a nice myofilament lattice localization of mitochondria, as you can see here, these mitochondria now become globular and coalesce, creating all these large blobs that you see here. And this happens when we downregulate mitophagy. So that is one of the consequences. So on one hand, we have, as expected, more mitochondria. On the other hand, also, we have disruption of the mitochondrial network. There are other consequences as well. There is increased ROS production upon mitophagy downregulation, leakage of calcium out of damaged mitochondria. There is collapse of the inner mitochondrial membrane potential that is used to create ATP. And as a result, these mitochondria create much less energy. So we have a phenomenon of mitochondrial aging, and a phenomenon of accelerated mitochondrial aging if we downregulate mitophagy. And as a consequence, these animals that exhibit lower mitophagy experience oxidative stress. And this creates the paradox I told you about earlier. The paradox is that although we have all these adverse effects upon mitophagy downregulation that I showed you, there is no effect on lifespan. And why is this paradoxical? Because we know that anything that can go wrong in a cell will have a measurable effect on lifespan. Lifespan is actually a very sensitive gauge of whatever goes wrong within a cell or an organism. because it's a cumulative phenotype. It is developing over time, over extended periods of time. So even the slightest perturbation will have eventually a measurable effect. But here we have a very steady, rock-solid lifespan under conditions of mitophagy inhibition. So we suspected that something must be compensating for this mitophagy downregulation that allows animals to not alter their lifespan. And with this, I will come to close to the end of my presentation. I will just present to you some of the data regarding this compensatory mechanism that we discovered. So, of course, you know that whenever cells experience oxidative stress, there is a signaling pathway called the retrograde signaling that results in the activation of the expression of genes encoding mitochondrial proteins in the nucleus. Most of the mitochondrial proteins are encoded in the nucleus, and the expression of these genes is under the control of the master regulator PGC1 alpha that works together with a number of cofactors of the NRF family, shown here, and this results in the upregulation of mitochondrial biogenesis. So whenever cells sense that their mitochondria don't function very well, they initiate this pathway to create fresh functional mitochondria through the process of mitochondrial biogenesis. Now in C. elegans, we don't have a PGC1 alpha master regulator, but we have a transcription factor called SKIN1. This transcription factor is a member of the cofactors, of the family of cofactors shown here, NRF family of cofactors. And we decided to test whether or not this transcriptional induction pathway cycle is actually involved in compensating for mitophagy downregulation. So indeed, what we find is SKIN1 becomes activated upon mitophagy downregulation, and in turn, it activates the transcription of target genes that encode mitochondrial proteins. As you can see here, these are just some examples. We performed microarray analysis of all mitochondrial proteins. These are some proteins involved in the respiratory chain function. Indeed, SKIN1 upregulates the expression of mitochondrial proteins upon mitophagy downregulation. But interestingly enough, it also upregulates the expression of DCT1, which is a mitophagy receptor, is the specific mitophagy receptor that I described to you earlier. So we have here two phenomena happening. On one hand, SKIN1 upregulates mitochondrial biogenesis. On the other hand, it also upregulates mediators of mitochondrial turnover. So this suggests that it co-regulates both processes. Now, if we don't have this compensatory circuit that is mediated by SKIN1, then we see the true effect of mitophagy downregulation. And this is my last data slide. So if SKIN1 is not there to perform its compensatory function through activation of mitochondrial biogenesis, then downregulation of mitophagy will result in severe lifespan shortening, as you can see here. In the double mutant, SKIN1, DCT1, for example. But we tested other combinations as well. I'm not going to go into the details in the interest of time. And finally, to summarize what I've told you, this is what we discovered in C. elegans. It's actually an evolutionarily conserved pathway that we now know also functions in mammals that regulates two opposing processes. On one hand, mitochondrial biogenesis that creates fresh functional mitochondria, and on the other hand, mitophagy. How is this happening? Whenever cells sense that there is accumulation of damaged or superfluous mitochondria, there are two pathways that are initiated. The first one is the oxidative stress response pathway, which results in the activation of this master regulator, SKIN1. Now, SKIN1 performs a dual function. On one hand, it activates mitochondrial biogenesis by activating the transcription of mitochondrial protein encoding genes in the nucleus. On the other hand, it also activates the expression of DCT1, which is a mitophagy receptor. However, although this is required it's not sufficient to induce mitophagy. To induce mitophagy, we need to now modify this DCT1 that has been synthesized through the pink parking pathway. And this is a safety mechanism that allows cells to not cannibalize the fresh mitochondria that have been generated. So instead, the pink parking pathway is only activated in the vicinity of damaged mitochondria. And this, of course, results in the induction of mitophagy, specifically taking down damaged mitochondria and leaving the healthy, fresh mitochondria intact. And now this process is also interfacing with other signaling pathways. The insulin IGF-1 signaling pathway is one of them, but it's not the only one. For example, we know that caloric restriction, dietary restriction, lowers insulin signaling and extends lifespan in part by inducing mitophagy. How is this happening? It's happening because dietary restriction lowers insulin signaling. This results in the repression of SKIN1, which is repressed actually by insulin signaling. So this results again in the activation of this pathway, mitochondrial biogenesis, and the transcription of the mitophagy receptor. Now, DCT1 is also under the control of another transcription factor, DAF16. It's a FOXO-transcription factor that is also repressed by insulin signaling. So low insulin signaling de-represses SKIN1, allowing for mitochondrial biogenesis and enabling mitophagy. On the other hand, DAF16 is also repressed, specifically inducing the expression of DCT1. And that is how mitophagy becomes induced under low insulin signaling conditions or under dietary restriction, caloric restriction, to extend lifespan in model organisms and to also protect, in some cases, cells such as epithelia and the hypodermis. And as I said, this pathway is also operational in mammals. It has been also identified in the mouse. And there are now specific ways of modulating this process that controls essentially the balance between two diametrically opposing functions, mitochondrial biogenesis and mitophagy. And it might be an entry point for interventions that allow manipulation of mitophagy in a pathology context, especially in cases of skin diseases that are associated with defects in mitochondrial homeostasis, as I showed you in the first slide. And with this, I will finish by thanking my colleagues and friends at the Medical School of the University of Crete in Heraklion in Crete. Specifically, Kostas Palikaras and Irini Ionachi were the two people who did most of the work that I showed you today. And I would be happy, of course, to take any questions that you may have.
So, time for questions. So, I have two questions. So, just to beginning. So, in this previous slide, so there is a link between oxidative stress and overexpression of PIK1 or not?
So there is actually a pathway. I'm showing it here as a summary. There are several proteins that are involved between here to here. There are also signaling events that result in activation of PINK1 expression upon oxidative stress. So PINK1 also is upregulated upon oxidative stress, similarly to parking, actually. So both of these proteins become upregulated under oxidative stress.
And the second one, just before the people question, so is it possible to modify glutathione homeostasis in C. elegans?
Yes. So indeed, we studied glutathione metabolism and homeostasis, and this also modulates mitophagy. So
the ratio between oxidative and
yes, indeed. So there is a connection between glutathione homeostasis and mitophagy, and if it becomes, if glutathione homeostasis is not maintained, then we have induction of mitophagy.
Thank you for your talk. One of the greatest contributions that C. elegans gave us for the aging field is actually the tissue-specific complementations, that we know now that it's not required to have this gene function in all parts of the animal, but in specific tissues. And you just showed another example of that. I was wondering if this has now been recapitulated in vertebrates. Do we know if mitophagy is more important or sufficient in specific tissues than others?
Well, not to my knowledge, there is a short answer. I don't know that this has been done specifically, tissue-specifically in the mouse, for example, or other vertebrate models. In C. elegans, we've examined, in addition to epithelia, to the hypodermis, we examined muscles, neurons, intestinal cells, secretory cells, and indeed we find that there is a pronounced impact on the hypodermis. And epithelial cells more generally appear to be particularly sensitive to perturbations of mitophagy. But I don't know if this holds true for mammals. Maybe it has been done, but to my knowledge, I don't know.
And another curiosity would be whether it interacts with the IGF-1 pathway in terms of the gonads to brain to intestine.
Yes. This is a very interesting question and topic more generally. We are actually now doing experiments to address exactly that. So to address cell autonomy versus cell non-autonomy in these responses. So what we find is that induction of mitophagy, for example, in the intestine, will result in the induction of mitophagy in other tissues that are not under stress. So we now try to identify how this is happening. Is it happening through endocrine signaling, perhaps, or other types of signaling? And this is true for other signaling events in C. elegans more generally. So, for example, specific mitochondria unfolded protein stress in neurons will induce mitochondrial unfolded protein stress in the intestine through a neuroendocrine axis. This is something that has been studied and we know. But we don't understand yet how mitophagy is regulated in a cell non-autonomous manner.
Thank you.
Question from the audience? Yeah.
So I have a question about the aging mitochondria. How could you explain that these aging mitochondria are less efficient than fresh mitochondria? And are you able to evaluate the production of ATP or energy?
Yes. Actually, we have now methods to quantify the production of ROS from mitochondria, calcium that leaks out of mitochondria, and ATP. So we can measure ATP being produced by mitochondria. We can even discriminate using optical reporters between healthy and damaged mitochondria in cells. So we know, for example, that this mitochondrion, which is red, is damaged, whereas the other that is green is not damaged. So there are tools now to monitor ATP production, ROS production, calcium leakage out of mitochondria, and overall mitochondrial health. And this allows us to do this in vivo. In C. elegans, for example, C. elegans is transparent. So it's very easy to put animals under the microscope, alive animals, so you don't need to sacrifice them, you don't need to prepare tissue samples and do electron microscopy. You can do this in vivo using high-resolution confocal microscopy. And that way you can even monitor ATP production in specific cell types. There are now reporters that allow for monitoring ATP production, for example, in the hypodermis or in neurons. This is something that we can do in the lab. We can also do this at the level of the whole animal. So how much ATP is produced by a specific C. elegans animal that is of 10 days of age versus 6 days versus 1 day. So we can quantify ATP production across lifespan. And this allows us to dissect these processes And of course, to answer your first question, how this damage is happening, it is happening because most of the reactive oxygen species actually damages proteins of the respiratory chain, oxidizes proteins of the respiratory chain in mitochondria. It also generates mutations in mitochondrial DNA. And this is another whole field that I have not really said anything about. But mutations in mitochondrial DNA will also result in deterioration of mitochondria because mitochondrial DNA encodes specific components of the respiratory chain that are very important. So since mitochondria are replicating in cells, if there are mutations in DNA, these are inherited by new mitochondria that are generated through fission, for example. And this, of course, results again in further problems, further deterioration of mitochondria function.
A question from just before Anika. So, two of your C. elegans colleagues got a few months ago the Nobel Prize.
Yes, Victor Ambros and Gary Ravkun. Yeah, I know them both very, very well.
I know that you probably have questions just after mine. So, did you check also microRNA that could control all this pathway?
We haven't. The short answer is we haven't really touched upon microRNA involvement in these processes, but it's very interesting. Actually, we are planning to do this in the near future, if we have the resources, if we find the people to do it. Then
Anika, maybe the question.
Thank you for the interesting presentation. Well, you showed that these mitochondria, they accumulate during aging, and then you showed these genes which are involved in the process, but maybe I missed that, but do the expression of these genes change during aging? So what is the reason for the mitochondria accumulation during aging, and can you utilize this knowledge to rejuvenate the cells?
Yes. So indeed, how is this failing during aging? What is happening? So expression of mitophagy regulators declines with age. This is something that we measured. And DCT1 expression goes down, parking expression goes down, PINK1 expression goes down. The question now becomes, why is it going down? What regulates this down regulation during aging? We find that it is epigenetic drift that is regulating this. So we have epigenetic changes. So these are stochastic epigenetic changes that happen because of damage that becomes accumulated in master regulators of these genes. Epigenetic changes that happen due to life history. And this results gradually in the downregulation of the whole pathway. So this is actually more general. It does not concern only mitophagy. General autophagy goes down during aging. So other types of autophagy also go down during aging. And this, of course, adversely affects mitophagy induction and allows this accumulation of mitochondria. On the other hand, because mitochondrial biogenesis is under repression, as you can see here, if this goes down during aging, which it does, this de-represses SCIN1 and allows activation of mitochondrial biogenesis. So you have a more widespread deregulation during aging. So things go down, but on one hand, mitophagy goes up, and then mitochondrial biogenesis also can go up, because if insulin IGF-1 signaling goes down during aging, then this allows for the repression of SCIN1 and increase of mitochondrial biogenesis. So there are both processes contribute. On one hand, you have mitophagy going down. On the other hand, mitochondrial biogenesis goes up. And that exacerbates the accumulation of mitochondria during aging because of this epigenetic drift that happens in the master regulators of these genes.
So do you see any possibilities to reverse this process, to rejuvenate the cells? Now we come back to insulin, which is a very, very popular topic nowadays, of course, with metformin and also the Ozempic and so on. Would this impact this process, or do you see some other ways to to intervene with this pathway?
So yes, for example, we all know that caloric restriction extends lifespan, and this is a surefire way to extend lifespan in almost all organisms. Actually, from yeast to even humans, it has been experimentally tested in primates, in monkeys, so we know that this far, caloric restriction is a sure way of extending health span and lifespan. Now, of course, this happens in part because of low insulin signaling, because that's what's going on. If you don't have enough calories going in, then insulin signaling will go down. And this also results in mitophagy induction and mitochondrial biogenesis. So results essentially in a more healthy pool of mitochondria in cells. And this explains in part why you have this longevity, this lifespan extension. Now, can you reverse these changes that happen during aging? There are some reports that suggest that you can reset the epigenetic clock, not in the context of mitophagy. This has not been studied for mitophagy specifically, but as a phenomenon, the epigenetic drift that happens during aging can be reset, for example, by using, by expressing the so-called Yamanaka factors in mammalian cells. But in C. elegans, I don't know of any experiments that have replicated this. So it could be, in theory, done if you had a way to reset the epigenetic clock or keep it ticking at a more decelerated pace. These stochastic phenomena will happen, but perhaps we can intervene to postpone them or decelerate the rate at which they are happening. Could be possible, but I don't know.
Okay, thank you. It's a promising prospect to prolong life without keeping away from the French cheese.
Yeah.
Thank you.
Well, you don't want to overdo it because, you know, I mean, if you intervene, for example, to reset some of these changes, you can cause carcinogenesis, you can cause cancer. For example, we all know about telomeres and how telomeres can contribute to aging of replicating cells, like skin cells. But actually, keeping telomeres long might result in cancer, in the immortalized cells that will be cancerous. So there is a thin line, and I don't think that we understand enough yet to really manipulate these processes in a secure way, let's say.
Last question.
Yeah. So thank you very much for this very nice presentation. You said that there is a relation between insulin and mitophagy, and it is known that there is some common phenotypes between aging and diabetes. So I have a very naive question.
Yeah.
Is it possible to produce some diabetic C. elegans?
Yeah, so of course we have C. elegans models of diabetes that can be studied, and there are mutants that affect the insulin receptor, for example. DAF2 is a gene encoding the insulin receptor that recognizes insulin, and we have ways of manipulating the activity of the insulin receptor in C. elegans. Also, the downstream cascade, the downstream kinase pathway that results in the phosphorylation of transcription factors that mediate the effects of insulin signaling. This we can do. But the interesting thing is that medications that have been approved for diabetic people, such as metformin, for example, which is a known drug, a known pharmaceutical to treat pre-diabetic conditions, actually extend lifespan in C. elegans. And this has been shown in many other systems. So metformin now is even in clinical trials in the United States. It is administered to healthy people, not diabetic people now, but healthy people. And what this clinical trial is about is to assess whether or not metformin can affect health span, not so much lifespan, because that would need, following these people for many, many years. But whether or not metformin in healthy people can improve health span, can improve biomarkers of aging. And this is ongoing right now, together with other medications that have been used in diabetes, in the context of diabetes. Now these are used as anti-geriatric drugs because they have been found to extend lifespan in C. elegans. For example, rapamycin is another such compound. Resveratrol, you may...
Urolithin too also?
Huh?
Urolithin?
Oh, yes, of course. Yeah. Also, urolithin is even more relevant to this story because urolithin is a specific inducer of mitophagy. It has been found that it is a specific inducer. If you want to induce mitophagy specifically, yes, inducer. So if you treat animals with urolithin A, you extend the lifespan of these animals because you induce mitophagy. Yes.
Other?
Yeah. Thank you for sharing this here in the back row.
Ah.
Hello. Thank you. Thank you for sharing this nice data. And so if I understood correctly, the ultimate reason behind the disturbed mitophagy during aging is the epigenetic drift. You surely are aware of the results from the Sinclair lab, who suggest that the underlying reason behind the epigenetic drift is DNA damage. So my question is that would there be a link between the DNA damage and the disturbed mitophagy? And what do you think about this whole theory between DNA damage and the epigenetic drift?
Yes, I mean, DNA damage is pleiotropic. It can do lots and lots of things. It's not just one thing. Of course, one of the things that it does, I'm not arguing against this hypothesis, one of the things that it does is that it affects epigenetic modifications. So that has been shown even in yeast. So it's not disputed. Now, whether or not it's a causative to decline, to age-related decline, this we have to really examine it further because there are also mechanisms that repair DNA damage. And these mechanisms are highly effective up to some age. So what happens afterwards and how these two pathways cross-talk between them, I don't think it's known yet. But of course, there is a link between DNA damage and epigenetic modifications, and it could be part of the reason why we have this epigenetic drift that results in the deregulation of all these processes in the cell.
Thank you.
Okay. So, thank you so much, Nektarios, for your presentation.
Thank you, Lionel.